In this lecture, we're going to talk about transcriptional regulation. So this is the actual production of a gene product from a gene. Now, a little bit of history. We actually, at least in bacteria, we didn't know that, or it used to be thought that bacteria just expressed all their genes at a constant rate. They were all constitutively expressed at the same time. And that they lacked the ability to adapt to their environment. And it wasn't until the 1950s that someone, Torriani Guarini, discovered alkaline phosphatase, which was expressed specifically on phosphate starvation and not expressed at normal times. So that's where we learned that bacteria actually adapted to their environment. So we now know that the expression of virtually all genes, even genes in bacteria, are tightly regulated. Think about it from a evolutionary standpoint. Why would this be the case? Well, proteins are one of the most metabolically expensive things for the cell to produce. So if you don't need an enzyme, for example, or you don't need a transport protein for a particular nutrient because you've got other nutrients that are available that are better, you can garner more energy from, then why would you express it? You don't want it to be active. Okay, so why would you even express it? You'd only express it when you needed it. But once you've expressed a protein, you want the ability to turn it off or turn it on when you need it. You don't want to have to make new protein and then destroy it when it's not used. Now, it turns out regulatory controls, there's multiple structural genes that get expressed at the same time, all from the same, on the same mRNA. Okay, so there's actually a subtler level of control that tells the ribosome to translate this message or translate the first message more than the next message on the mRNA. And that way you can further regulate the expression of genes. So if you need 2 copies of one gene to 1 copy of another gene for whatever purpose in the cell, you can actually put a translational control in between those 2 structural genes that are co-expressed on the same mRNA. So there's a level of translational control as well. So we have 3 levels where we control the activity of a cell, the proteins that are produced by the cell. The first level is transcriptional, and this is where we're producing mRNA from a structural gene, presumably so that mRNA can be translated into protein. And then once the protein's produced, because we paid such a metabolic price to make it, we can allosterically, through some ligand, regulate the activity of that protein or that enzyme. Or we can actually covalently modify that protein to regulate its activity. Allosteric regulation is a more subtle regulation where it adapts to varying levels of some regulatory ligand, and there can be multiple controls on that protein, multiple allosteric controls on that protein. Post-translational modifications are more like a switch. It's on or off. And all of these are discussed in the book. There's a whole section on allosteric and post-translational modification of proteins in the chapter on enzymes. We're not going to discuss that here. Here we're going to just look at transcriptional controls or transcriptional regulation. All right. So even at the transcriptional level, there are 5 possible stages of transcriptional control. We can repress transcription so that no mRNA gets produced from that structural gene. We can de-repress it, where the gene is now constitutively expressed. So there's nothing repressing it anymore. The gene would just be transcribed at a rate consistent with other genes that have no regulatory control. So there's repression and de-repression. Furthermore, we can activate. So we can, there can be a regulatory protein that binds ahead of the structural genes on the DNA that actually recruits RNA polymerase to come and transcribe that gene. So we get an enhanced production of the gene product in the case where it's activated. And that can be separate from the protein that represses it. Matter of fact, it usually is. Now, there's a more subtle level of control where the transcription can be amplified. And we're not going to talk about that in this lecture. There's some great examples in the book where that occurs, and that often occurs where the regulatory proteins themselves are regulated by themselves. So the expression of the regulatory protein is controlled by the regulatory protein itself. And that gives you really unique ways that things can happen. So you can get into autocatalytic amplification, autocatalytic production of some gene product where the regulatory protein controlling that is under its own control. And the faux regulon is a great example of that, and there's a whole chapter devoted to the faux regulon. There can be amplification just from production of different sigma factors. Okay, at different phases of the of cell growth, different sigma factors. Sigma factors are part of the RNA polymerase, and they direct the RNA polymerase to different structural genes in the genome. So there can be different sigma factors, and there can be anti-sigma factors that tie up those sigma factors so that they no longer will bind to the RNA polymerase and direct its activity. So there are ways to amplify both the magnitude of the response to whatever is regulating the gene. You can amplify the rate at which the gene is produced. You can make the gene more or less responsive to higher or lower concentrations of that whatever is triggering the stimulus, or whatever is triggering the response. So that's beyond the discussion here. There's a whole section devoted to that with examples in the book. All right, but at some point you have to be able to turn off the response. So the last possible stage is attenuation, and this has often been overlooked in the literature. Okay, if you think about a system that has no attenuation, you immediately think of viruses. Okay, viruses just keep producing more and more and more copies of themselves inside the cell, and they never turn off, and because of that, they eventually lead to lysis of the cell, and the cell dies. Okay, so without attenuation, the cell can overproduce stuff, to the point where the cell bursts. So there has to be a way to turn off the response or attenuate the response. So these are the 5 stages of transcriptional control. Not all of these stages are demonstrated by every structural gene. Most of these occur for a lot of structural genes. So let's look at how this happens. Okay, so let's look at constitutive gene expression. Now, constitutive gene expression is where there's no regulation of that gene. The mRNA polymerase just finds the structural gene on the protein as it's moving around on the DNA, and it says, "Oh, I can transcribe this. Let me go ahead and transcribe it." And then, as it moves on, another mRNA comes along and says, "Oh, here's a free structural gene. Let me go transcribe it." Okay, so constitutive gene expression is the easiest to understand. I have a gene, it just gets expressed at a constant rate. Okay, so we're ignoring any translational changes. We're basically assuming that we're in some sort of balanced growth. So the rate of gene product accumulation now is the concentration of the gene product, and whether you're thinking of that as protein, if you ignore any translational changes, that could just be the protein that's produced from the structural gene after the mRNA is produced, or it could just be the messenger RNA. So we're just going to call it generically a gene product. But the concentration of the gene product is the concentration relative to the biomass, total biomass. So if we multiply the concentration per unit biomass by the total biomass, X, then we get the total amount of gene product produced. And we're accumulating that over time. Okay, and the rate of production is proportional to the number of genes coding for that gene product. Again, this is genes, there's a concentration of those genes times the biomass. So we have Cg, which is the gene concentration per unit biomass. X is the total biomass. All right. So we can write a material balance, which you see over here to the right. And we're looking at the total accumulation, rate of accumulation of the gene product with time. And that's proportional to, over on the far right, that's proportional to the constitutive rate of expression of that gene, Kc, times the total amount of the total number of genes that are present. Now, we can take that derivative and factor it. So X times dc, the change in gene product concentration with time, plus the gene concentration, or the gene product concentration, times the rate of change of biomass with time. Well, we know from our growth models, okay, that the rate of change of biomass is equal to the specific growth rate mu times the amount of biomass that's present. Okay, so we can make that substitution and then rearrange this expression and solve it for dCp dt, and we get a fairly simple expression. Now, this is the rate of production per unit biomass, so it's the constitutive rate of expression times the concentration of the gene, and this second term is the continued dilution of the gene product as the biomass grows. Okay, so we have two competing generation terms. We have one that's generating new gene product. We have another that's diluting out the gene product that's been produced based on the growth rate. Because as we accumulate new biomass, we have to be producing, we'd either be diluting out the protein or the mRNA that was produced, or we have to produce more to compensate for the biomass. So we have those two terms. Now, what happens if mu goes to zero? Well, if mu goes to zero, we just keep accumulating biomass. That's bad. That's what a virus does. Okay? So this is why most genes are tightly regulated. And you don't want to have something that's just constantly on, just keeps accumulating gene product, because at some point you fill up the cell and then you burst the cell, if the cell is not growing. Okay, if the cell continues to grow, you're okay. All right, you can, as those two need to be balanced out, but you can get to a point where dCp dt goes to zero, and we'll talk about that in a minute. All right, so during balanced growth, mu, the specific growth rate, is constant. The rate of constitutive gene expression should be constant. It depends only on the amount of RNA polymerase that's around and how well RNA polymerase transcribes that gene, that structural gene, and the concentration of structural genes per unit biomass. So you could have multiple copies of a gene, particularly if you're putting a gene on a plasmid. You can have multi-copy plasmids, and hence you have a higher number of copies than in the cell, but in exponential growth, which is a form of balanced growth, cells usually have about 2 to 2.5 copies of their genome around. So you could have 2 genes per, 2.5 of structural genes per copy per cell at any given time. If you have plasmids, you could have even more. You might have 10 to 20 copies around in the cell. All right. So we can integrate, since all of those are constant, the only thing that's changing is the gene product, concentration of the gene product. We can integrate that expression starting from whatever the initial concentration of the gene product is and go to the gene product at any point in time. And what we see is a constant term and a transient term. Now the transient term tells you how fast you're approaching a steady state, because at long time, e to the minus mu t at long time goes to e to the minus infinity. This term goes to 0. Anything multiplied by 0 is 0. So we end up with a constant. So as long as we're in balanced growth, the steady state level of the gene product should be a constant. And we can calculate what that is. So if we know the steady state level of an mRNA, if we did quantitative RT-PCR, we can calculate how much mRNA is being produced from any given structural gene. And at that point, if we know the total number of copies of the structural gene in the cell and we know our growth rate, we can actually calculate what the constitutive rate of expression is. Now, that's cool. We can actually measure those kinds of things. Okay, so there is a way to get at what is a constitutive expression rate. As I said, though, this is a very simple model, but most genes are not constitutively expressed. Okay, so we really need to look at a regulated gene model, and we'll talk about that in a second. First, let's talk about how transcription is regulated. Okay, so here I've got a stretch of DNA, and this is the stretch of DNA that is ahead of, it's about 110 bases ahead of the first structural gene in the lac operon. Okay, that's just where I'm getting this. So it comes from the lac operon, but it's not the structural gene of the lac operon. It's a region of DNA ahead of that structural gene. In fact, we know where the mRNA is initiated, and that's at this point in that sequence 0. Okay, and I'm calling that's the 0 point. So I'm going minus 70 back from that 0 to plus 40, or a little beyond plus 40, and you'll see why after that 0. But this is where the mRNA actually starts. So I call that my 0 point. And I can count my bases from 5' to 3' from that point. Now, I'm only showing the sense strand. Remember, there's a Watson strand and a Crick strand. DNA is double-stranded. Okay, I'm only showing one of the two strands because you know that the antisense, if this is the sense strand, the antisense strand can be determined from the sense strand. Okay, now, it's actually not the sense strand that gets produced, okay, or gets transcribed. It's the antisense strand, because you want the mRNA to be the same sequence or the appropriate sequence to the sense strand. So here we have the antisense strand, okay, and I'm just showing it right where the gene, the protein would begin. So this is the beginning of the structural gene, and it's about plus 37 from where the mRNA starts. Okay, here is where this is the mRNA that would be produced, and remember, so T becomes uracil in mRNA, so the sequence is the same as the sequence in the sense strand. So I'm actually coding. This is the code that would be read by the ribosome. This is the code. This is the equivalent DNA for the mRNA that's being produced. Now, this is the initiation site of the protein. Okay, so almost all proteins start with methionine. Not all, most start with methionine. Okay, so we have the first three amino acids now, methionine, threonine, and methionine for the first protein of the lac operon. It turns out that's the first three amino acids of lac Z. Okay, so this is the translation initiation site, is this AUG codon. But this is how it relates back to the sense strand and the antisense strand and the mRNA in the genome. Now, I said 0 is, or actually +1 from 0, is where transcription gets initiated. Well, it means I've got a region here before the structural gene starts that I could put something, if I wanted to, into the sequence. And I've got a region ahead of this that I could put something else. Now, how does the cell actually know where to start transcription? Well, it turns out Shine and Delgarno worked that out, and it's called a TATA region, and TATA is the consensus, but it's a thymine and adenine rich region in the DNA, and it's these short green codons here that are roughly -7 from the transcription initiation site, and -35 from the transcription initiation site. So this is where the RNA polymerase binds to the DNA. Okay, and it's called the Shine-Delgarno sequence, or the TATA region for short. All right, so this is where the RNA binds, and it knows that the RNA polymerase binds, and it knows to start in this region from that binding site. Now, I still have a lot of room between where the transcription starts and where the actual structural gene starts. I could fit something in there. I could let something else bind there. And in fact, there's a binding site there for another protein. Turns out the LAC-I protein binds to this sequence in the DNA. Okay, now notice that binding site, think of the protein now as covering the DNA, covering the double strand of the DNA. Okay, and it's actually overlapping where transcription initiation would start. So I stands for inhibitor. It's that whenever a protein binds here, transcription is inhibited. It's stopped. All right. So there's reasons that there's kind of non-coding regions in the DNA. This is the reason. I can have another protein binding here which would repress transcription of the structural genes that are downstream of it. Now, we've also got this region up here. That's a fairly big region. There's no structural genes in that region. Well, I could fit another binding site there, and indeed there is one. Okay, there's a binding site for the CRIP protein, and CRIP is an interesting protein. Okay, CRIP is part of a regulatory scheme that shows when the cell is under catabolic stress. So when CRIP binds here, CRIP actually recruits RNA polymerase to the Shine-Delgarno sequence, and it makes the RNA polymerase bind there more often, and hence amplifies the production of the downstream genes. Now, look at the order. CRIP is ahead of the Shine-Delgarno sequence, and it recruits RNA polymerase, but if LAC-I is occupied with a inhibitor protein, that RNA polymerase does nothing. Okay, so the inhibitor in this case is dominating the response, and that becomes important. Now, I'd hate to be looking at all this sequence and trying to find stuff. Okay, that's just nuts. All right, so we usually write this in a shorthand. Okay, and this is the shorthand. So we've only shown the first 3 amino acids, or the first 9 codons of the LAC-C structural gene. The gene goes on for quite a long way. Then after that, there's the LAC-Y structural gene and the LAC-A structural gene. Those are the 3 genes that are part of the LAC operon. And we call that an operon because all 3 genes are expressed from the same Shine-Delgarno site. That's this arrow at +1. That's the transcription initiation site. There's a LAC-I box, or binding sequence. We call that a box. After the initiation site, there is a CRIP box for amplifying production of those genes ahead of the Shine-Delgarno sequence and the transcription initiation site. So this is just a shorthand way to represent the entire LAC operon, so we don't have to be staring at ATCGs forever. All right, so this gives us something we can look at. Now, we can add to this the LAC-I protein, which is binding to the LAC-I box, and this is an example of repression, derepression. If that protein is not bound to the DNA, we get constitutive expression. Okay. If CRIP binds and LAC-I is not bound, then we get activation or promotion or amplification of those downstream, the expression of those downstream gene products. Okay, if both are bound, the order of precedence becomes important. If both are bound, nothing gets expressed. The gene expression is repressed. Okay, it's only active when LAC-I is off of the DNA. In other words, it's inactive, and then it's derepressed and constitutively expressed or actively promoted when CRIP binds. So we have to get the order of precedence correct in order to do the model. All right, so I've explained it in pictures, what we're looking at. What's the math behind this? Okay, so let's look at a generic situation. We have a single regulatory protein. I'm going to call it just a regulatory protein because if it's a repressor, the regulated rate at which K-regulated, the rate at which that structural gene is expressed, would be less than that it would be under constitutive expression or unregulated expression. Okay, if it's activated, then if it's an activator protein, then the rate of expression would be greater than constitutive expression. Okay, that's how we tell the difference between repressors and activators. And a lot of times we think of repressor proteins as the rate of transcription goes to 0. It might. Okay, it doesn't necessarily mean the rate of translation goes to 0, but it certainly slows it down over constitutive expression. All right, so we only have one regulatory sequence, or one box to which that regulator protein binds. The regulator protein can be active when it's bound, or it can be inactive, and we'll talk about that in a minute. But that means the gene takes on 2 states. The gene can either be regulated or it can be constitutive. Constitutive always exists. Okay, it's a question of how much of the gene is regulated that really determines the rate. Whether it's up or down depends whether it's an activator or a repressor. All right, but we have a total concentration of the gene. Now this is again concentration per unit biomass. It's not concentration in a batch. Okay. All right. So the only difference between the expression from a regulated gene and constitutive expression is the rate constant that's associated with the transcriptional rate from the gene. Okay, so we can write a material balance around our gene product, and that the gene product is changing over time. That's the rate of accumulation of gene product, and it's equal to the regulated rate of transcription times the concentration of the regulated gene plus the constitutive rate of expression times the concentration of the unregulated gene. Okay, minus any growth and growth dilution of the gene product. Now, the regulator protein, once it's bound to a sequence, you can't bind a second. Okay, there's only room to bind one to that box. There can be other regulatory boxes that can bind the same or other regulatory proteins. Okay, but in this case, once that box is occupied, it's occupied. You don't bind a second. So the binding of the regulatory protein to the DNA is an equilibrium. And we can define that equilibrium constant as the concentration of the regulated genes is equal to or divided by the concentration of the constitutively expressed gene. That's what that would be an open box times the concentration of the active regulating protein. That's what CRA is. All right, so we've got building up a simple model here. Now, we have to think about how is that protein, that regulatory protein, being shifted from active to inactive. And there are two ways to activate or deactivate the regulatory protein. One is by allosteric regulation with a binding ligand, and that's actually the way the lac operon works. So if this is the repressor or the activator, so it's either lac-I or CRIP in the lac operon, there is an allosteric regulation. Lac-I is allosterically regulated by lactose or galactose, and CRIP is allosterically regulated by the alarmone cyclic AMP. Okay, so both of those are examples of allosteric regulation. The faux regulon is actually a covalent modification. So they're two different things, and these lead to two different models. We discuss both these models in the book, but in this case, we're just going to look at one. Okay, and we're going to look at allosteric regulation. And I can do this two ways. I can either bind a ligand to the inactive to make it active, or I can bind a ligand to the active to make it inactive. And I've just arbitrarily assumed that the ligand binds to active regulator to deactivate the regulator or make it inactive. And now I can write an equilibrium expression for that in terms of the concentration of the regulatory ligand, L, and the two states that regulated protein could be in. Now I can write a material balance for the regulated protein. The total amount of regulated protein has to be the sum of the active and inactive versions of that regulated protein. Again, all of these are concentrations per unit biomass. And if I plug that into and do the appropriate algebra, I can write the concentration of the active protein is equal to the total concentration of the regulator protein divided by 1 plus KLCL. Okay, so now I have a way, if I know the total amount of protein, I can predict how much is active based on how much regulatory ligand is around. All right, so if I substitute that in for the gene product model and integrate that, because I'm assuming we're in balanced growth, so the total concentration of the regulatory protein, okay, CT sub R, is constant. That's definition of balanced growth, is everything is constant in the cell. Well, the total is constant, but the active and inactive may not be. Okay, the total amount of gene is constant. That's CT sub G. KL is a constant. K equilibrium R is a constant. Okay, so all of these are constants. I know Kr and Kc are constants. They're my rate constants of transcription. So if all those are constants, I can integrate now what is Cp as a function of time. Okay, I can integrate that expression because everything is constant. And what I see are two transient terms. Okay, if I have an initial concentration of the gene product, that's going to end up disappearing with continued growth. Okay, and that's the kind of dilution effect for continued growth. If as time goes to infinity, e to the negative infinity is 0, this drops out. We have 1 minus e to the negative mu t. As time goes to infinity, this goes to 0, and what I end up with now is a constant. So at steady state, Cp still goes to a constant, but that constant now depends on the concentration of the regulatory ligand and the total concentration of the regulatory protein. Now, this is a complex. Here's the steady state equation. This is a complex equation. There's a lot of terms to deal with. So let's dimensionalize this. Let's take this, put this into some sort of dimensionless form. What we see is Kl, Cl always appears together. Okay, well, Kl has units of 1 over molar, or 1 over concentration. Cl is a concentration. So L, if I define L as Kl times Cl, that's a dimensionless number. Okay, well, I can let R equal Kr times Ctr because those two terms always appear together. Okay, again, this has units of reciprocal concentration times concentration, and I'll call that R. That's a dimensionless number. Okay, if I divide through by the total amount of genes, that cancels this term and puts my concentration of gene product per unit of gene. And both of those have units of concentration, so P is a dimensionless number. Now I'm left with Kr, Kc, and mu. Well, it turns out Kr and Kc are rate constants, but they have units of reciprocal time. Mu has units of reciprocal time. So if I divide Kc and Kr by mu, if I took this into the numerator, I can divide Kr and Kc by mu, and I can call that K and K prime respectively. Those are dimensionless numbers. They're dimensionless rate constants. Okay, that means I can get the steady state dimensionless concentration of the gene product per unit gene as a function of L, R, and the two rate constants. That's a simple equation. I can look at that now, and I've solved it for any values of Kl, any values of Kr. I can do a dimensionless graph of that, and if I were to assume that R was an activator protein, so transcription is activated when Ra goes up. Okay, and I can do that for different concentrations of that activator protein, or dimensionless concentrations. Then you can see that at low concentrations, I get a little bit of boost in activity as the ligand drops off. As the ligand goes up, this decays because I'm driving it to the inactive form. Okay. If my activator protein goes up, I get more. As the concentration goes up more, I get more, and so on. So as I keep building up the concentration of the activator until I get up to the point where it's equaling the genes, then my transcription is activated as I lose regulatory ligand. As I gain regulatory ligand, the transcription from that gene drops back to 1, which is the constitutive expression rate. All right, so that's if R is an activator. What if R is a repressor protein? Okay, if it was a repressor protein, The active protein is repressing the gene, the expression of the gene. Inactive is allowing the gene to be transcribed. And here we can see the same thing. As long as I have a high concentration of the repressor relative to the binding equilibrium constant, as I increase the regulatory ligand concentration, I get more and more expression. As I decrease that regulatory ligand, I get less and less expression. And it depends on the relative concentration, the relative dimensionless concentration of that regulatory ligand as a repressor. This is, and if you look, all these curves are sigmoidal. And that's what we expect to see, is kind of a sigmoidal response around the point where the regulatory ligand is, the concentration of the regulatory ligand is approaching the equilibrium constant, or the reciprocal of the equilibrium constant. And that's where the transition starts to occur. Okay. So it depends whether we have an activator or a repressor. This is a very simple model of a single regulatory ligand. You can have multiple regulatory boxes in front of a set of structural genes. The models can get very, very complex. You can even have binding boxes between genes, between structural genes of an operon. Okay, and they can either activate or they typically can repress. If there's only a single Shine-Delgarno sequence, they can repress the expression of downstream genes, only allow the expression of upstream genes. The Hut regulon in, or the Hut operon in Bacillus subtilis is a great example of that. And there's a whole chapter devoted to the Hut operon in the textbook. So there's lots of ways that cells have adapted to use these regulatory boxes. There's both global regulation schemes like CRIP and cyclic AMP, where whole sets of genes get turned on or turned off due to catabolic stress in the cell. There are gene-specific regulators, where a gene only gets turned on under certain conditions and may be like the lac operon. You have to have lactose around before the genes required to metabolize lactose get produced. Okay, so without that, those genes are unnecessary and they're just turned off. They're not produced. So that's a good example of the regulatory control depends on something being present. All right, so this is an introduction to a whole series of chapters in the textbook that talk about these regulation schemes, both allosteric regulation and covalent regulation. The regulation can occur at a single gene level, at an operon, which is a collection of genes back to back with under a single Shine-Delgarno sequence, or a regulon where there are concerted sets of genes all regulated by the same regulator, either repressed or activated by the same regulator, but they are disparate in the genome, and each operon of that regulon has a Shine-Delgarno sequence in front of it, so they can be handled independently. And that good example of that is the phosphate or faux regulon in E. coli. That's been studied for 70 years. The study continues on that. It's very complex, and they're still not totally worked out. Okay, but we now have a very, very good model starting from some work that was done about 30 years ago. We now have a fairly complete model for the faux regulon, and that's in the textbook. Okay, so that finishes this lecture. Again, I hope this intrigues you to look at the book and buy the book, and you can go much further than what I can do here in a single lecture. Thank you.